Hello, welcome to my study. I'm Peter Fox, Principal Investigator of the BrainMap project. BrainMap was first introduced to the OHBM community 30 years ago, and it's been widely used ever since. In this presentation, I'll be introducing you to our latest creation, the BrainMap HPC Community Portal. BrainMap HPC is a digital ecosystem designed to help researchers map neural networks involved in normal task performance, as well as networks afflicted by network-based disorders. Both objectives are achieved by meta-analysis of the coordinate reporting neuroimaging literature that we all contribute to. This is the homepage of BrainMap.org. This site provides access to the BrainMap databases and BrainMap applications, and has for some time. There are three BrainMap databases. The Task Activation database contains task control contrast experiments acquired with fMRI, PET, and other functional imaging methods from both healthy controls and patient populations. The voxel-based morphometry database, on the other hand, contains case control contrast of structural MRI images, localizing disease-associated alterations. The voxel-based physiology database is the newest release, being debuted here in Montreal at OHBM 23. Like the VBM database, it contains case control contrasts, both of hemodynamic and metabolic alterations caused by diseases. The BrainMap applications, Scribe, Sleuth, GingerAile, interact with the databases. Scribe codes data for entry, Sleuth performs filtered data retrieval, and GingerAile performs coordinate-based meta-analysis using activation and likelihood estimation. Jointly, they provide meta-analytic maps using a mass univariate approach that is a type of statistical parametric mapping. As pervasive as statistical parametric mapping has become since it was first introduced more than 30 years ago, it has one substantial shortcoming as regards brain function. Maps are not networks. They're spatial patterns, but there's no intrinsic connectivity information in them. And we've known for approximately 400 years, starting at least with Descartes, that the brain is composed of functional networks. Charcot and other neurologists reporting lesion deficit cases in the 1800s intuitively and correctly interpreted their findings in terms of networks. Since the 1980s, and continuing to the present, numerous strategies for network modeling have been developed. Van Essen used electrophysiology in primates. Biswald developed functional MRI and functional MRI connectomics. A crucial challenge for neuroimaging meta-analysis is whether network models can be derived from statistical parametric maps, that is, from static maps. Can we get out connectivity information? A widely cited finding that really never ceases to amaze me is shown here. Steve Smith performed ICA analyses on the BrainMap task activation database and in parallel on 35 healthy volunteers. In the BrainMap meta-analysis, shown here as the BM meta-analysis, each reported study is the analytic equivalent of a single time point in a resting state fMRI time series. The networks extracted for each method, BrainMap, task activation, and resting state, are remarkably similar. The physiological inference is that resting brain continuously exhibits the same networks, although more subtly, that are evoked by task performance. The methodological implication is that meta-analysis can extract network properties as latent variables from an assembly of statistical parametric maps which individually have no network information. An extension of this strategy to brain diseases is shown here. The columns labeled functional ICA are a replication of the Smith ICA analysis I just showed using the task activation database, but with more total data. This is more recent. The columns labeled VBM ICA are the same analytic method, ICA, but performed on the voxel-based morphometry database. That is, on disease-related alterations across a wide range of neurologic and psychiatric disorders. The pathophysiological implication of these networks emerging is that brain pathology recapitulates healthy physiology, at least at the network level. Otherwise stated, the majority of chronic brain disorders exhibit network-based degeneration. This is an unexpected extension of William Seeley's network degeneration hypothesis, which was motivated by a small number of neurodegenerative disorders, particularly tauopathies, which definitely demonstrate network spread of the pathological proteins. The same strategy, but with a different analytic approach, has been applied by Nick Crosley that's shown here. Crosley used graph theory modeling rather than ICA, but like Smith, Crosley compared BrainMap task activation data with resting state fMRI and found very similar topological properties both at the whole brain level, shown on the left, but then also at the subnetwork level, shown on the right. Crosley then extended this work to a comparison of disorders, here comparing 20 different neurologic and psychiatric disorders, again drawn from the BrainMap databases, here from the BrainMap VBM database. The upper right panel illustrates the network degenerations in Alzheimer's disease, shown in the purple, and schizophrenia, shown in the green, and their overlap in lavender. So Alzheimer's and schizophrenia, which are largely anatomically distinct, do share some hubs. The larger conclusion is to again reconfirm that many disorders exhibit network-based degenerations. And so we want to develop methods to see these network disorders, to model the network disorders. And the voxel-based morphometry and voxel-based physiology literatures provide an excellent way to do that. A final example of meta-connectivity modeling strategies is shown here in this slide by Robinson et al. This shows the meta-analytic connectivity modeling, or MACM approach, that applies the AL algorithm to map connectivity as patterns of co-occurrence with activations in a seed region. Here the amygdala is the seed, and the co-activations throughout the brain are connected networks. For validation, this was compared to the COCOMAC database of amygdala connectivity in the rhesus. It was highly similar. So this is a third metaconnectomic approach and a third validation. The MACM approach has recently been extended by Eitan Kotkowski, who developed a pipeline approach to using AL to discover nodes, both in health and disease, and then using MACM to identify edges to make a more formal node and edge model out of the connectivity maps that Robinson generated. Here he's modeling network connectivity in the amygdala, and this was done both with structural data, healthy control data, and with voxel-based morphometry data. The next step in this approach has been recently reported by Florence Chang, who used this disease node and edge modeling strategy to develop a resting state functional MRI biomarker of gray matter alterations in multiple sclerosis. A patent's been filed for this, which is pending now. But the approach is shown here in this pipeline is that the VBM and VBP databases are used to provide disease nodes as a disease metamap. Edges are provided through MACM, and then this model is formalized using structural equation modeling and can be then validated out of sample in primary data. This also has been published by Chang. So from these preceding examples, I've shown that there are multiple metaconnectomic methods that can be applied to task activation and disease-based data. The chief impediments to bringing these into general use are that they require a large amount of literature data, they require pipelines that are not straightforward to implement, and they require high-performance computing. So collectively, this is the motivation for the BrainMap HPC portal. BrainMap brings together the BrainMap databases, connectivity pipelines, and a science gateway high-performance computing infrastructure in a way that allows both users and developers to add to the environment and perform validations in primary data. So this is a complete ecosystem for metaconnectomic modeling and connectivity biomarker discovery. The portal architecture is illustrated here. This three-tiered design of topware, middleware, and deepware has been honed by the Texas Advanced Computing Center over several years and is very robust and user-friendly. It provides resources for both the non-developer, shown here, the researcher, and for developers who can make new tools and get direct access into lower levels in the system. The starting point for access is portal.brainmap.org. Once you enter this system, you get access to the applications and the data. So in this presentation, I don't intend to do a detailed walkthrough, but you can see that ICA, MACM, graph theory modeling, connectivity-based parcellation, and author topic modeling are already implemented with other connectivity applications in development. So that's all I wanted to show you today, but the entire BrainMap team and I invite you to explore, enjoy, and contribute to this new research resource, the BrainMap HPC portal. Thanks for your attention.